So hello there, my name is Nigel French, and it's a real pleasure to be able to give this presentation at the conference. I'm going to be talking about the epidemiology for One Health, and in particular I'm going to be talking about some tools that we've been using to inform how we control and prevent infectious diseases at that interface between humans, animals and the environment. And a word about One Health first of all. So One Health is really a recognition that there is an inextricable link between human health, animal health and environmental health. And there are many issues related to public health and also to animal health that fall within this domain. And these are four of the ones that we are particularly focused on in New Zealand in an organisation, a group, a centre called One Health Aotearoa. And these are issues related to antimicrobial use and resistance, environmental pollution, in particular how it relates to public health and water safety, and also food safety. So a number of pathogens that come from animals affect people through food. And lastly, but extremely importantly at the moment, is the emergence of infectious diseases at the human-animal environment interface. And COVID-19 is a very good example, ongoing example of how important this area is. Now, my talk is going to focus on water safety, food safety, and some environmental health issues. And I'm going to illustrate the techniques I'm going to talk about, mainly using examples from New Zealand, mainly because these are areas that myself and my group have been working on for a number of years. But just first of all, a bit about water safety. So waterborne diseases are extremely important public health concern globally and still estimated to cause over 1.5 million deaths annually, linked to things like poor sanitation, hygiene, and environmental pollution. And there are many pathogens that are involved in waterborne illness, both bacterial, protozoa, but also viral illnesses as well that can be contracted through water. So this is a global One Health concern, particularly affecting developing countries, but it's also important in New Zealand, and I'll explain why in a minute why this is important. We have both sporadic cases of infection, but also some very large outbreaks, and I'll illustrate one of these later. Now, the techniques that I'm going to be describing a bit more in detail are techniques around the area of source attribution, source tracking. And these are really important for identifying the animal reservoirs, the sources of infection for people so that you can put measures in place to control them. Now, this is also important for food safety, and we know that foodborne illness is also an extremely important cause, particularly in developing countries, particularly in children. We can see that there's a large issue with this, particularly with food safety and foodborne pathogens in resource poor countries. But also it's important for countries like New Zealand that depend a lot on our economy for international trade of food and food products. So it's important for industry as well as for public health. Now, One Health, this is again animals obviously infecting the food chain and people picking up infection through the food chain is very much a One Health issue. And source attribution, again, identifying the source of infection is extremely important for informing public health policies. Now, just another point about the pathogens that cause foodborne illness. Many of these also affect people through environmental contamination as well. So this could be contamination affecting families or rural families in particular, people living in rural areas, and also occupational exposures too. Why is this important, particularly in New Zealand? Well, we can see the average, if you look around the world, the ratio between livestock and people is around about 2 to 3 to 1. So about 2.5 livestock kilograms per kilogram of person. Whereas if we look at in New Zealand, we have extremely high populations of livestock. And in fact, the ratio in New Zealand is around about 30 to 1. And this explains why zoonoses, foodborne, waterborne, and environmental zoonoses are an important source of infection pressure to our population. Now, in New Zealand, I mentioned this is the list of notifiable diseases reported to our Ministry of Health. And we can see that at the top of the list is campylobacteriosis, still measured at a rate per 1,000, But there are other important zoonoses in there as well: Giardia, Cryptosporidium, Salmonella, Yersinia, sugar toxin E. coli, and right at the bottom there, leptospirosis. So many of these are important food and waterborne illnesses. So source attribution, what do I mean by source attribution? We use source attribution to estimate the relative contribution of different sources, and I'll define what I mean by sources, to the burden of human illness. And this is where a lot of the newer techniques and modeling approaches can be used to inform policy for prevention and control. So how many infections are coming from chickens, how many are coming from wildlife, how many are coming from ruminants? So it's important for both surveillance and also monitoring of interventions in food and water chain, and also for preventing both sporadic cases but also large-scale outbreaks. And again, just to underline that because we're looking here at humans, animals, and the environment, it requires that One Health approach. So what do I mean by sources? Here I'm going to describe them as reservoirs, pathways, exposures, and risk factors. And this is just one way of looking at this. So we can define the reservoir as the animal host or the maintenance host that's actually amplifying the pathogen of interest, be this Salmonella, Campylobacter, or Leptospira, or whatever. So these are the animal hosts. And then as we move nearer to the point of consumption by the human, we can then see that these must go through various pathways. And these can be important for determining what government agency may be responsible. For example, there may be a separate agency responsible for food compared to water or environment, and also for occupational health. Then as we get nearer, we can think about particular exposures. So these that are nearer to the individual point of consumption, and then even more refined, we can look at specific risk factors. And I've got an example on the right-hand side here, where you might have a cattle as a reservoir contaminating the food chain as a pathway. The hazard is the milk supply, or the exposure is the milk supply, and the particular risk may be the consumption of raw or unpasteurized milk. Now, this is a framework described in this paper here, just to illustrate how there are various different techniques that we can use. So reservoir attribution modeling all the way through to things like case control studies, and I'll illustrate the combination of use of attribution modeling with case control study later on in the talk. And these inform, so when we understand the pathways, the exposures and risk factors and the animal reservoirs, that can inform policy for reducing infection. Now in New Zealand, Campylobacter is a really good example to illustrate this. This is a foodborne, waterborne and environmental pathogen. And between 1980 and 2006, we had an increasing incidence, which was really much higher than many other OECD countries. So this graph here illustrates how poorly New Zealand was doing, even allowing for reporting biases compared to other countries. In 2006, we had over 16,000 cases, confirmed cases, which was really high rate and unacceptable rate. And this paper was published in 2006, really saying, well, we think it's chicken that's the most important source of infection. We really need to control this to reduce the epidemic. But at the same time, there was still uncertainty around precisely what the cause was. Was it cattle? Was it wildlife? Was it chicken as the source? Earlier work had implicated poultry as a source of infection, but there was still, the evidence was considered weak. For example, this case control study here identified eating poultry at a friend's house was increased risk, whereas eating at home was protective. And this was considered not really strong enough evidence to put strong measures in place to control what was happening in the poultry industry. So we had a rising incidence, but still some uncertainty around what the precise sources were in New Zealand. So in 2005, the Manawatu region, which is this region here in North Island of New Zealand, was set up as a sentinel surveillance site. And samples from human cases were coming through the local public health laboratories through to our laboratory in the Massey University in the town of Palmerston North here. And there we sequenced those isolates at the time just using 7-gene multi-locus sequence typing, a particular strain typing method. We also isolated from poultry supply, from other food sources, and also from high-use recreational swimming areas, and also farms in the catchment areas around there too. So we had simultaneously information on the types that were coming through people, causing illness in people, and those that were coming from food and from the environment and from wildlife as well. So for the first time, we had a really good picture of the strain types of Campylobacter in New Zealand that were causing illness in people and how they were related to the animal reservoirs. And I'll point to a couple of interesting ones here, which I'll pick up again later. Sequence type 474, this one here, almost unheard of anywhere else in the world other than New Zealand, but strongly a cause of human illness and also associated mainly with the poultry supply earlier on. In other ones, we had ones like sequence type 2026 here and sequence type 61, again, important causes of human illness, but identified mainly in ruminant sources. We had the two species, jejuni and coli, both important causes of illness in people. So you see that many of these occur not exclusively in one source, so you have to use some modelling approaches really to identify what the relative importance of the different reservoirs are. Now in around about 2004, 2005, I think it was, there was an early work that was developing a particular model, which is called the asymmetric island model, which was published by Danny Wilson. And this combines a kind of population genetics evolutionary modeling approach with sequence typing, in this case the 7-gene typing, to model both the evolution of the pathogen and also the relative flow of genetic material from these individual reservoirs into the human population. And it was that relative flow that was used to determine the source of human infection. Now, there are many other models that have been developed, and some of them have been developed by our team here in New Zealand and published. Different features, different approaches. Some of them use genetic materials, some of them don't use the genotyping information at all. Some of them you can include other variable information. So, for example, this model here, which is source R, available as a CRAN, as a R package in CRAN. It's a fully joint model. It allows covariates such as age, severity, and rural-urban classification to be included in the model. And anyone who's interested, there's a review of all the different types and models of source attribution that have been described. These were 2 papers that were published last year that are good sources of information on the different types of model. To cut a long story short, when I applied these various different models to the data in New Zealand, we could identify that indeed poultry was the main source of human infection. And actually it was one particular poultry supplier that was the cause of most of the infections in our sentinel surveillance site, and subsequently we were able to confirm this was a national situation as well. This resulted in a risk management strategy in the poultry industry. A number of different measures were included. The industry worked together to try and reduce the level of contamination on the poultry carcasses for sale in supermarkets. And the effect on public health was very dramatic and very quick, and I think much quicker than we'd anticipated at the time. So, for example, we can see the relationship here between notifications and hospitalizations after the introduction of the strategy. We saw a 50% reduction in both notifications and hospitalizations attributable to campylobacteriosis. So this was a very successful intervention informed by the source attribution modeling. That 50% decline was also resulted in a significant saving to the New Zealand economy through people not requiring health interventions, but also not taking time off work either to look after themselves or look after sick children. And we could see that the decline was attributable to poultry. We use our source attribution modeling. We can see that the decline in human cases was largely due to poultry-associated genotypes. This is a model that was published just last year, an extension of the asymmetric island model and another model by comparison. So this is a Dirichlet distribution model and the island model at the bottom here, where we've added covariates to look at the relationship between attribution and things in this case, such as the spectrum where you are from rural to urban. We have a scale of rurality in New Zealand, and also before and after the intervention. And we can see that if you live in a rural area, you're more likely to get infected from Campylobacter from ruminants, whereas if you live in an urban area, it's very similar between the two models, you're more likely to be infected with poultry. And that declined to some extent after the intervention, which is what you would expect to happen. Now, that was just described using different modeling approaches, some of them using genetic data, some of them not using genetic data, adding covariates in. But that was all based just on 7-gene multi-locus sequence typing. With the advent of whole genome sequencing, we now have a much more powerful approach to look at, in this case, at particular genotypes as well. So I mentioned earlier there was one sequence type 474, which was the cause of 1/4 to 1/3 of the human cases, particularly early on in the epidemic. Predominantly one poultry supplier, and early studies revealed it was rapidly evolving, particularly through recombination rather than substitution or point mutations. And we were interested in how and why this particular strain emerged in New Zealand and what caused it to decline afterwards. So we have genome sequenced all of the isolates of sequence type 474 that we had in the collection over that time period. And what we could see was a lot more detail. So if we just looked at the 7-gene scheme, all of these isolates would be the same. But now we can see actually they fall into 4 different clades when you use whole genome sequencing. These 2 clades are present all the way throughout the epidemic. This clade was in one particular poultry supply, and this was the cause of the first epidemic wave between 2005 and 2008. Then we saw a major evolutionary bottleneck associated with the intervention in the poultry supply and the emergence of a new clade, which has not been isolated since 2014. So we've seen this background groups here, and then the emergence of new clades. Now what we've done is applied our asymmetric island model to looking at source attribution now using whole genome sequencing. And just to illustrate the power of whole genome sequencing, if we had just used our multi-locus sequence typing data on our 474s, we'd have no way of identifying what the potential animal reservoirs of the individual human cases are. If we have the 7-gene scheme and we have other multi-locus sequence types, we can say, well, we don't know for individual isolates where they were from, but it's most likely that they were from this particular poultry supplier. But once we have whole genome sequencing, we have much higher resolution, and we can begin to see that these clades that were around throughout the whole epidemic were actually more associated with ruminants, whereas the ones later on were much more associated with the poultry supply. And when we map the attribution back to the clades, we can see a strong association with the epidemic group and supplier A. We see this escape to a post-intervention clade, again associated with that group, and then both of these disappear. And in the background is this consistent picture of ruminant-associated sequence type 474s over that time period. Now this leads to a number of different hypotheses, but one of them is that 474 potentially jumped over from the ruminants into the poultry supply, caused a big amplification, and that's what led to a major increase in human cases. The evidence for this is that it belongs to a group associated with ruminants, and the evolutionary analysis and the attribution supports a jump from ruminants to poultry. Now that was one particular strain, and then in 2014, after that particular strain virtually disappeared from poultry, we saw the arrival of a new strain, and we don't really know exactly where this came from, but the difference between this one and previous strains is this was resistant to 2 classes of antimicrobials. We first detected this in our Sentinel site, and it was found in all the poultry companies and in the breeder parent flock. So a very rapid dissemination across the poultry supply, only in North Island of New Zealand. We saw a big increase in resistance as a result of this emergence of this clade in this particular sequence type in human cases and in poultry at the same time. And it was also responsible for a major outbreak in Wellington, food-associated outbreak. Genome sequencing showed that this strain was new to New Zealand. It was very different to anything that had ever been here before. So we were interested to say, well, how long has it been in New Zealand? How has it managed to transmit so rapidly around our poultry supply, and why has it emerged to become such an important source of human infection? and how is it evolving. And really we needed whole genome sequencing, and we needed a One Health approach because we needed to sequence from humans and from the poultry supply at the same time. So we used whole genome sequencing. We used both long read and short read sequencing of isolates from humans and from the different poultry supplies. And we could see that even though it seemed to appear relatively recently, it was already revolving into different clades that were associated with different poultry supplies in different regions of New Zealand. This is a group here associated with this part of New Zealand, and this group here associated with this part of New Zealand. The red circles are the human cases, which really show that human cases are associated with all of the different poultry supplies that we had in New Zealand at the time. So we're interested in finding out how it was transmitted so rapidly around the poultry industry in North Island. Why was it spread so quickly within companies? Is there evidence of local spread? Is it spread through contact networks? We know that poultry farms are connected through a network of personnel, of feed, of chicks, of other sources as well. So this work involved looking at things like the genetic distance. So we could look in great detail at the whole genome sequencing and look at the genetic distance and how that relates to things like road distance or Euclidean distance. And we can see this is a tanglegram which shows the relationship between companies, but also genetic distance and road distance. So already we're beginning to see that there may well be evidence of both local spread as well as within company spread. But in order to do this in more detail, we used more genetic modeling. In this case, we used a multivariable model based on distance-based redundancy analysis, looking at the different networks. In this case, the genetic network or the genetic phylogenetic network, but also how that relates to things like the feed network, the live bird network, hatching eggs waste, but also road distance and Euclidean distance. And what we could find when we did these multivariable models is that the genetic distance and by inference transmission was closely associated with the parent company, individual farms, road and feed networks, particularly important in that analysis. So we could answer these questions through genome sequencing and through modeling. How long has it been in New Zealand? We can estimate a common ancestor of around about mid to late 2013, which implies that's when it may have entered New Zealand. We still don't know how. It seems to be spread within company, and there's evidence of local spread, possibly through road network and the feed network. All companies contributing to human infection. And if anyone's interested in the genetic basis for resistance, this is a paper published in EID at the end of last year that describes that in more detail. Now, just a couple of other things I want to just add before I finish. One of them is this particular incident here. So up until now, I've been describing infections which are most likely to have come through food or through environmental sources, so ruminant-associated or poultry-associated infections. This particular spike here in 2016 was one of the largest outbreaks, of waterborne outbreaks of campylobacteriosis ever recorded in the world. In August 2016, and this was associated with contaminated groundwater in New Zealand, in the town of Havelock North, which is on the east side of North Island. It's a shallow aquifer. This is an example of a bore well on this particular road here, and the population of Havelock North only had 15,000 people. There was a heavy rainfall event, and then followed by a classic epidemic curve, point source curve. It's estimated over 8,000 people, so that's more than half of the population of the particular town affected, going down with campylobacteriosis as a result of the contaminated water supply. 42 hospitalized, 3 developed complications of Guillain-Barré syndrome, 4 died, complications associated with campylobacteriosis. Certainly the biggest single instance of waterborne disease in New Zealand, and one of the biggest in the world. Large-scale economic loss as a result of the outbreak, but also serious public health issues. So it was associated with a rainfall event. The water supply was not chlorinated, it was untreated, so people got sick, but then chlorination was introduced once the peak of cases occurred, and that resulted in a rapid decline in infection. This is described in a recent paper, if anybody's interested in this detail. Now, genome sequencing and modeling was important for understanding the source of this particular infection. Although we knew it was the groundwater, we had multiple different whole genome sequence types involved in the strain. And when we look at core genome multi-locus sequence typing, so using the whole genome data, we can see that there was a very strong association between outbreak cases, the reticulated water supply, and also bore water, but also sheep feces from sheep that were grazing in the contaminated area. So the most likely scenario was a heavy rainfall event, drainage of sheep feces into the shallow aquifer. As the water was untreated, a large proportion of the population got seriously ill. So in this case, a large outbreak associated with contaminated water supply. The last thing I'm just going to mention is source-assigned case control studies. This is an approach which combines both source attribution modeling and also the classical case control study design. And there are a couple of other studies which have been published using 7-gene multi-locus sequence typing. We combined whole genome sequencing with source attribution in this particular study that was completed over 2018-2019. And the basic design is that you have your cases and your controls, but you assign your cases according to the genotypes from the human cases into the various different potential sources. And then you analyze poultry-associated human cases or cattle-associated, in our case, human cases, using the same control set to identify specific risk factors. So over 600 cases, 600 controls. We also had isolates from various different sources, from poultry, cattle, from sheep, and we were able to identify risk factors associated with poultry-associated infection and cattle-associated infection. To do the source attribution, we looked at a number of different gene combinations. So these are 3 different combinations of genes. This is the typical 7-gene scheme that I mentioned earlier on, but this is a 52-gene scheme, and this is another scheme. Using source attribution, we can see that there's reasonable consistency between those different approaches, that a large proportion of the cases were poultry associated and a large portion of the remainder were cattle associated. Now, if you're interested in the details, there's a publication here which is going to be submitted for peer review very soon, which describes in more detail the risk factors associated with this. But essentially, we could see that there were poultry-associated risk factors and also cattle-associated risk factors. When you just analyze the cattle-associated cases, you get a very strong association with things like the consumption of raw milk, working on a farm, and working with animals. So we can see the difference, much higher resolution when you start looking at cases associated either with poultry or with cattle. So just to finish the last slide here, so obviously food, water safety, security, these are important One Health issues and some of the biggest challenges facing the population today. Food production not only is a potential source of foodborne illness, but also can contaminate the environment and water supplies and can have a direct negative impact on public health as well. Identifying the source of these infections and transmission is extremely important for putting in place measures to improve public health and to mitigate against these infections coming through those different pathways. And it's helped by new tools, both whole genome sequencing, but also advances in modeling, and this is continuing ongoing work which is improving our approach to this. And I mentioned earlier, these are what I've described are very much One Health approaches at the human-animal environment interface. And that's just the last slide, just identifying the many people and the funders that were involved in the work that I've just described. So thank you.